Hey everyone, today we're going to be looking at how DNA recombinant expression works, its impact on research, and improving human life in general. To start, this technology is one of the most amazing and versatile research techniques that biochemists have been developing since the 1970s. Recombinant DNA is created when you combine DNA from two different genetic sources. The most common recombination involves fusing a gene of interest from a plant or animal, including humans, into the plasmid of a bacterium, let's say E. coli, as shown in the image below. This practice is also called molecular cloning. But why would anyone want to insert DNA into bacteria genomes or create new DNA in the first place? Well, we can think of the bacteria as our workers that will produce the necessary protein for our purposes. Some useful applications will be to identify genes that cause disease using transposons in lab research, or to enhance the durability of nutritional value of crops, GMOs, or to mass-produce proteins such as the insulin hormone for treating diseases. Let's focus on this last application and go through the entire recombinant expression technique to mass-produce insulin. Okay, so it all begins with an isolated gene of interest. Here we use the polymerase chain reaction technique to make millions of copies of our first insulin gene. Once that's done, we need a plasmid vector to insert our PCR amplified gene into, and here's where the most important players come in. Restriction endonucleases create sticky ends at the ends of the gene of interest and in the plasmid. This happens at the sites labeled N and X by cutting the palindromic sequences there. With these sticky ends, just align the two ends and two X's of the plasmid and gene and use a ligase enzyme to glue in the gene. And here's our recombinant DNA. Not too difficult, right? Well, we're not done yet. How do we express insulin now that we made a recombinant plasmid? Let's grow an E. coli culture and plate it on agar. Add the solution of recombinant plasmid onto the plate. One more thing, add an appropriate antibiotic to the plate as our plasmid has an antibiotic resistance gene. This way the E. coli cells that have taken up the plasmid will survive by expressing this antibiotic resistance gene. Finally, we can pick a specific colony that has survived to cultivate further before inducing the gene expression system to produce lots of insulin molecules, ready to conquer diabetes. So, although this process seems simple, it is very powerful and biochemists remain motivated to ensure that the technique is regulated as safely as possible before publicly using it to solve our problems of healthcare and food sustainability worldwide.